Se está conectando.
Sí, no pasa nada. Hello. Hello. Good afternoon. Buenas tardes para aquellos que estamos en Australia. Muy buenos días para todos los que estéis en España. It's an honor to be here in Instituto Cervantes of Sydney to present Australia and Spain, the Symposium on breast cancer on behalf of the Awareness Month of breast cancer here with my colleague David Gallego Ortega. It is an immense privilege to be here with David. He is a professor in UTS, director from Single Cell Technology Facility from the School of Biomedical and Engineering. It goes without saying that Instituto Cervantes of Sydney, along with our fellow researchers, are working hard to promote science and science in Spanish. After the current director from Instituto Cervantes and former Minister of Science in Spain signed an agreement to promote science and science in Spanish, it is our duty from all Instituto Cervantes, more than 88 Instituto Cervantes around the world, to promote science and to promote science in Spanish, science in association, in our case, with our fellow researchers from Spain and our researchers from Australia. I would like also to thank la Asociación Española del Cáncer, as well as the National Breast Cancer Foundation from España, Spain, and Australia, also to be here with us. It will be David Ortega, who will be the maestro de ceremonias. He will be the presenter of all of these high-level researchers that will be able to give us an insight in this research, in the research of this topic. I would also like to acknowledge the traditional owners of the land in which we are meeting. I pay my respects to their elders past and present, and to the Aboriginal elders of other communities who may be here today. Without further ado, it is an honor once again to be here in Instituto Cervantes of Sydney with our fellow researchers and to be able to promote science and to promote science and research in Spanish. And once again, thank you for both associations, Australia and from Spain, to be here with us. David, the floor is yours.
Well, thank you very much, Coral. Really, I'd like to also extend my acknowledgement as well to Coral and the team at the Instituto Cervantes for hosting us today here for this symposium. I guess that I just wanted to really say that this is an example of, highlighting a little bit of the collaboration that happens between Australia and Spain and many other countries, but obviously we are focusing now in Australia and Spain because research and breast cancer research, there's no borders in research, and then we are all in the same kind of boat, try to investigate better cures and find better treatments for breast cancer. So today we have 4 speakers. So 2 of them are from one from the ASEICA, another one from the National Breast Cancer Foundation. And then we have 2 researchers that are going to be talking about the research that they're doing to fight breast cancer. So the first speaker is Tony Celia Terrassa. So he's a group leader of Cancer Stem Cells and Metastasis Dynamic Lab at the Hospital del Mar Medical Research Institute. And even he's a researcher. He's really here today representing ASEICA, the Spanish Association for Cancer Research. And I would like to give the honor to start this session today. And over to you, Tony. Thank you very much.
Okay, thank you very much, Coral and David. Can you see my presentation? So can you see my presentation?
Yes.
Yes. Okay. Great. Well, thank you. I mean, very much for the invitation, Coral, David. This is a great initiative from Instituto Cervantes and the National Breast Cancer Foundation. As David said, I'm also a breast cancer researcher, but today here I'm in the name of ASEICA to present you and to show you what is ASEICA and what we do for science from ASEICA, that is the Spanish Association from Cancer Research. And one of the main things that we want to do is to strengthen and to reinforce boundaries with Spanish researchers outside Spain. So this is a classic example of the things that we really want to do, and so that's why we are so happy about this invitation in the name of the whole board of ASEICA. So ASEICA is the largest Spanish association of investigators. So it's an association for cancer researchers. We have more than 1,200 members at the moment, and we do different activities for them, especially for young researchers, for women, but also for the cancer research in general. Here I have a picture and a structure of the board of ASEICA. I don't want to go through details, just mentioning the president right now is Luis Pazares, and the next president next year will be Marisol Soengas. So we do a lot of activities, and we really want to put together the basic translational and clinical research of cancer in Spain. So is an association that we have clinicians and we have translational researchers and basic researchers. So this is very nice because we are building a network there that we put all together, all the phases of cancer research together in order to move forward. We do a lot of activities that I'm going to explain throughout the presentation. And we break down in different programs. So we have a program for ASEICA Woman and a program from we call ASEICA Aprende. That is, we are launching some courses, official courses in the university for oncologists or for researchers that want to learn more about translational research. Another program is ASEICA Youth that Maria Cafarel, that is giving the talk today, is also coordinating with myself and Veronica Torrano. ASEICA Med, that is a program for clinicians, also to attract more clinicians to the association. And then ASEICA with Society, that we do multiple things to basically keep cancer research in good shape and care about funding and employment in Spain. So about ASEICA Woman, this is a very active program. They do a lot of activities. One of the most famous activities, I would say, is the one called Conócelas, that is called the Cancer Detective. So once a year, a lot of women researchers across Spain give presentations to primary schools. So this reaches more than 1,100 students in Spain, 1,100 students in primary school, and it's an extremely successful program. But this is an open program that, all kinds of activities favoring this part of science is getting bigger and bigger every day. So about ASEICA Youth, that is the program that we are more involved, and one of the programs that we care the most, as I said, is coordinated by Veronica Torrano, Maria Cafarel, and myself. We had to establish a hierarchical structure because There is a lot of participation of many young scientists. So we have a working group beside the three of us. We have a working group of 15 young scientists that are also helping us to coordinate ASEICA Youth or ASEICA Joven. And then we have working groups or different sub-programs inside ASEICA Joven that we launch as activities. We have a mentoring program for ASEICA Joven and ASEICA Mujer. We have a program for PhD students, a program for postdocs, and a program for early PIs. So the activities that we do in ASEICA Youth are these: so mentoring, peer mentoring, networking, scientific sessions, and also training courses. So about the mentoring, we launched a program 2 years ago that was very successful. We had more than 50 couples, mentor-mentee, and that means that we have different levels of mentoring. So we have postdocs mentoring PhD students, early PIs mentoring postdocs, and also senior PIs mentoring junior PIs. And this is ongoing, and this is something that it's basically an online format of mentoring. So if any of you, even from distance from Australia, are interested, you can participate just sending us an email. And then we have not only this vertical mentoring, we also have peer mentoring, or more horizontal mentoring, that is a platform for PhD students, postdocs, and early PIs that they gather together periodically, so they can basically put together their concerns, their most interesting things or news, and help each other. So building also a network of confidence with other PhDs or postdocs or early PIs across Spain, but also many of them, especially in the postdoc group, are from Spanish researchers outside Spain, because this is only an online format. So again, all these activities, you can participate from the distance. So networking. This is an example of networking. We are extremely excited to have the first meeting of ASEICA Joven next month, because ASEICA Joven was started just right before the pandemics, so we couldn't gather together, and now it's going to be the first in-person meeting in Santiago Compostela in a month. So we are going, all these groups that I've been talking about, the PhDs, the postdocs, and the early PIs, will gather together in an event in Santiago Compostela next month. We will take advantage of this. So this is a general ASEICA activity, and it's the international meeting. So ASEICA is organizing an international meeting every 2 years, and it's going to be also in Santiago Compostela. And other activities for networking, an example is this one from ASEICA Med. Also, just right after the symposia, there will be a retreat for clinical oncologists, but also basic researchers will be there, just to put together in a more informal way the research that they do. We also do science sessions. We did this, for example, in collaboration with ECUSA from United States, Españoles in USA. That was called the Seed and Soil Program. It was an online format. Again, this was also during the pandemics. And we put together basically early PIs from Spain with postdocs in United States to present their research and make connections, because we were matching also the field of the researchers, the early PIs and the postdoc. So to put together the research and also to give advice on how you can go back to Spain if you want to go back, or other alternatives. but it was a very successful program, and we did more than 6 sessions, and maybe we'll do it again in the future. So we also have participated in other symposiums from Spanish organizations outside Spain. For example, the FERFA Symposium, that is the Association of Researchers, Spanish researchers in Germany. We do have some connections also with EMBO, and hopefully we'll start some activities soon. And we have also participated in other meetings, like the SEBBM, that is the Sociedad Española de Biología Molecular. And also we have participated in the EACR meeting in 2021 and the early career EACR meeting in this year in 2022. We also do this series of seminars that we call Meet the Expert. So we get some expert of some topic invited once a month or once every 2 months to have a bit of, to give us a flavor or a view from their perspective, from their trajectory in the field, and how they reached the objective that they reached. And this is with the aim to have an informal conversation with that expert. So we basically reserve like half of the time of the presentation just for open question, just to chat with the experts, so PhD students or or postdocs can have an easy access to some leaders for the respective fields. We have, of course, people from Spain, from associations in Spain, but also editors, people in the industry and researchers, or in the young academy in Spain. So this is also a format that is very successful and very useful for young researchers to be able to get in touch with such leaders. And we also do training courses. We want to shape also the leadership skills of our LPIs. These are, skills that we normally don't get training of during our careers, and more are getting crucial as we climb up in our academic career. So in these courses, or in this case, the paper mill is the company that give us these courses. They are experts in this kind of training, and so they shape the roles as leader, how to set priorities, work-life balance, delegate functions, or how to manage conflicts. So we also have other training courses, but for the sake of time, I didn't mention. But we also have we have a continuous offer of training courses for our members, and we also have the ASEICA awards. So every year we give 2 awards. One is the Maskeun Trail from Alcoy. This is a running trail in the mountains of Alcoy, and actually is a very emotional race. We've been there running or walking, some of us, and it's very exciting to see an entire city running and giving the money for research. So they give us every year the money collected from Maskeun Trail to give a research award. And here you have some of the awardees. These are always for young researchers. And we also have, together with the FERO Foundation, the foundation that was started with the philosophy of José Baselga, and in order to promote translational research. So together with FERO, we have the FERO ASEICA Awards that we give one also every year, and is also for young researchers, normally researchers that come to Spain and they need to start their own lab. So these are awards that are extremely helpful to launch their careers. And just the last slide, just to engage and do a little bit of advertisement of the international meeting. This is going to happen next month. Registration are still open, so if you happen to be around, do not doubt to come and reinforce your connection with the Spanish researchers. We have a great lineup of speakers, not only from Spain. We have keynotes like Tomás Gajewski or Daniel Piper, but also other leaders in Spain, like Ángela Nieto is also one of the keynote speakers. So if you are interested, we'll be very happy to host you there. And that's it. I'm gonna leave it here. And if you have any question, any doubts, I'll be super happy to answer you. We also have here Maria Cafarel, that is also a part of the board, so we can probably both together solve your doubts.
Okay, thank you very much, Tony. So I think that we're going to have questions at the end of the 4 presentations. Thank you for your overview on the activities of ASEICA. I think that now is very clear, hopefully for the people here in Sydney and also for the people in Spain that are watching us, what are the activities and the important work that ASEICA is doing over the years. I'm a proud member of ASEICA for the last probably 15 or more years. And I think I've been following for so long, even from the distance. I think it's a great work that you guys do. So congratulations on that. And thank you very much for your presentation.
Thank you.
So we're going to move on into the next speaker. We have here with us in Sydney. So we're kind of going Spain, Sydney, Spain, Sydney. So we have Associate Professor Samantha Oakes with us. She is a researcher, a very successful researcher in breast cancer. But now she moved into the National Breast Cancer Foundation, where she's Director of Research and Investment. And she's also keeps her adjunct associate professor position at the St. Vincent's Clinical School at the University of New South Wales. And Sam is going to tell us about the activities of the National Breast Cancer Foundation. And she's going to give us an update on what the organization is doing for breast cancer research. Thank you.
I'm going to stand up so I can see my slides, if that's okay, David. I might just stand over here so I'll see the slide. Thank you very much, David. What he didn't say is that David and I have had spent a couple of years working together, so huge amounts of respect for you, David. And I hope I can give you a little indication about who the NBCF are and what we do, and hopefully we can help you in the future to fund your research. So the National Breast Cancer Foundation is Australia's largest national body funding world-class breast cancer research in this country. We invest around 22% of the entire national investment in breast cancer research in this country. Government contributes about 50%, and National Breast Cancer is the second largest funder of breast cancer research in this country. We do that entirely on community donations. We receive no federal government funding. So we rely not only on the community investment, but we rely on all of the researchers out there to make incredible impacts so that we can keep using that to raise more money for you. As Director of Research Investment at the National Breast Cancer Foundation, it's my role not to judge the grants, but certainly to open, design and facilitate our world-class breast cancer research schemes. Every year, our main research scheme is the Investigator-Initiated Research Scheme, where we ask researchers all throughout the country to apply for our scheme. And it's my role and my team's role, obviously, to allocate those research projects to several peer review panels, and we run independent peer review panels. So each research grant is put into a peer review panel that may be reviewed by 8 to 10 researchers and clinicians throughout the country. So we ask national experts to review those grants, and ultimately it's the final ranking of those grants together with the consumer input that we rely on heavily that ultimately have the funding models presented and approved by the board, and that's how we invest in research. So I thought I'd start with where we're up to in research, what our main vision is, highlight a few of the research projects and show you what the true impact of that research is. This is where we're sitting. I'm sure I'm not telling this audience anything, breast cancer is the second most commonly diagnosed breast cancer in Australia. And incidence of breast cancer, shown in that blue line there, puts very simply, is continuing to outpace Australia's population growth. Australia's growing, we're getting more cases, but there are a number of factors related to how we're actually seeing a rise in the breast cancer incidence. National breast cancer started in about 1994, and you can see since that time, we've seen a reduction in the mortality rate from breast cancer. In fact, the mortality rate from breast cancer has decreased by 43% over that time, despite the increasing incidence of breast cancer. Ultimately, research saves lives. Our contribution to the national research investment in breast cancer research has led to this improvement in outcomes for those diagnosed with breast cancer. But the reality is that pink line is still not at zero. 10 cases per 100,000 will still die of their disease. 3,200 women and men will die of breast cancer this year alone. That's 9 deaths per day. 9 per day. One woman under the age of 40 will die every week in Australia. So although that we're doing very, very well, unfortunately, we've still got a long way to go. Research has led the way, but we need to continue to invest in world-class breast cancer research if we're going to bring that down to zero. And ultimately, the single purpose of the National Breast Cancer Foundation, our single vision, is 0 deaths from breast cancer. And we will do that by investing in world-class breast cancer research in this country. So far, since 1994, National Breast Cancer Foundation has invested now this year over $200 million towards world-class research projects, almost 600 world-class research projects since 1994. As I said to you, this has led to improvements in the mortality rate by 43%. And we invest in research, breast cancer research, across the breast cancer research continuum, from prevention and risk. Obviously, we don't want people to get cancer. They're not going to die of it. Better detection methods. If we detect it earlier, they're going to survive. And of course, new and improved therapies for patients with those really hard to treat or advanced and metastatic breast cancer. So in the last year alone, and I thought I'd give you a flavour of some of the research projects that we've funded in the last year alone, we've invested about $12.4 million into 20 new world-class research projects. And they cross the entire breast cancer research continuum. We've got a project from Associate Professor Claire Sturzaker at the Garvan Institute of Medical Research, who's trying to develop a blood test using epigenetic markers to detect breast cancer recurrence. So we know that we're really good at treating those sort of first step cancers, but which ones return? And using epigenetics, Professor Claire Sturzaker is trying to work out a way we can detect the recurrence of that disease earlier. Tony Taganis down at Monash University is trying to use immunotherapy to improve the outcomes of those who are diagnosed with breast cancer. And I'm going to go into a really cool project very shortly that we're funding. And Ian Campbell, interestingly, is working on a very large pool of breast cancers. He's got a cohort of breast cancer, 6,000 breast cancer cases plus 6,000 normals. And what he's learned that for the 10% of breast cancers that are inherited through a family, so familial breast cancer, we know that about BRCA1 and BRCA2 mutations account for about 10 to 15% of those. But for about half of all familial breast cancers, we have no idea what the mutation is. So what he's doing is he's using next generation sequencing to try and learn whether there are other perhaps regulatory regions that are mutated that are affecting breast cancer, BRCA1 and BRCA2 outputs. And I thought I'd highlight, oh, sorry, that got very excited. I thought I'd highlight a couple of, go into further detail about a couple of really cool projects that we're funding in the last year round of the investigator-initiated scheme. Obviously, this audience knows that there's a proportion of breast cancers that are obviously triple-negative breast cancer, classed as triple-negative breast cancer, heterogeneous class of breast cancers that don't express the estrogen receptor, don't respond to targeted or hormonal therapies. Andrew Scott has used some preclinical work to actually discover that he can turn off this particular gene, PDGFCC, and what that does is it actually ends up in the re-expression of estrogen receptor on breast cancer cells, making them now sensitive to hormonal therapy. So his work in this particular project over 3 years will use novel drugs to turn off this particular protein in combination with hormonal therapy to see whether triple-negative breast cancers can be made more sensitive to hormonal therapy. Really cool stuff. It's a different way to look at triple-negative breast cancer, because all of a sudden, instead of trying to find a drug that will actually target triple-negative breast cancer, why not make it more sensitive to anti-estrogen therapy? Really cool project. Associate Professor Liz Carlton at the Garvan Institute, that David knows very well, is working on, and obviously these slides are made for a multitude of audiences. What she's done is she's used CRISPR technology to define, really highlight what genes are regulated in resistant breast cancers. And what she's found is there are a number of genes that are expressed in particular, or a number of cells that express a subset of genes that are responsible for the way the breast cancer cells, learn how to survive. So she's potentially discovered that these particular resistant ER-positive breast cancers may be sensitive to BH3 mimetics, drugs that reactivate cell death, basically. So she's using that in combination with CDK4/6 inhibitors and estrogen receptor positive cancers to see whether she can reactivate cell death in these resistant estrogen receptor positive cancers. Another amazing project. Again, repurposing a drug from another cancer system, like blood cancers, to see if we can overcome the really true problem in breast cancer, resistance to treatment. And then I thought I'd highlight a couple of really amazing outcomes that we've had from already existing funded researchers. This one's a really interesting project. So Brendan Kennedy, we had a number of different grant schemes over the years. We've got our current investigator-initiated research scheme. Dr. Brendan Kennedy, who's a researcher over at the Charles Perkins Institute in WA, got an innovator grant for a very small amount of money to start this work, and we've continued to fund him as well as his postdoc subsequently. Now, what they have done is they've used 3D printing technology to develop a customized probe to assist surgeons at the point of surgery. There's a real problem with patients with early breast cancer. With early breast cancer, of course, we want to try to maintain the quality of life for a patient. So if they've got a lumpectomy, you want to remove the cancer and leave as much of the breast tissue as possible. But of course, it's a balancing act. Get rid of the cancer, leave as much breast tissue. And at the moment, it relies on imaging and the surgeon's ability to sense of touch to try and find out where those margins are. We know, unfortunately, for 30% of patients, they'll be recalled for a second surgery, right? That's obviously not ideal. We don't want to recall patients for a second surgery. It's problematic because ultimately they won't have clear margins. So what he's done is he's created this amazing 3D printed probe, which can now detect the stiffness of cancerous tissue. And that is very different to obviously the normal soft breast tissue. And this particular probe now just shows you from the investment that we've made in these researchers, he's now attracted an additional $2 million from the Medical Research Future Fund, and this probe has been fast-tracked towards commercialisation by the United States FDA. Really amazing outcomes. And I thought I'd highlight another program that we've been investing in for a number of years. And these slides have not showed up well on the slide anyway. You'll get the point. So we've got another program called the National Breast Cancer Foundation Endowed Chairs. One of those researchers that we're supporting is Professor Shireen Loy at the Peter MacCallum Cancer Centre. Now, these endowed chair fellowships are designed to support researchers for long periods of time. These endowed chair fellowships are large amounts of money and collaborative funding with the host institution over 10 years. Now, Professor Shireen Loy has told us she's globally the top 1% cited researchers globally. She has said to us that she's remained in this country thanks to National Breast Cancer Foundation Endowed Chair Fellowship and continued to do her work. Now, a number of years ago, funded by other NBCF, another NBCF grant scheme, she discovered that there's a proportion of breast cancers, including triple-negative breast cancer and HER2-positive breast cancer, that have this infiltration of tumour-infiltrating lymphocytes. So you can see that here. Obviously, tumour-infiltrating lymphocytes, and here's a cancer without. So there's a proportion of these really hard-to-treat breast cancers that have a large proportion of lymphocytes. You know this, David, because you've been working in this space for a long time. She developed this pathological test, obviously, simply scoring the number of tumor-infiltrating lymphocytes, and she discovered that that particular test will now predict the response, interestingly, of those patients to routine chemotherapy. But of course, we now know that immunotherapy is revolutionizing the treatment of several cancers, including metastatic melanoma. And of course, this has led her interest in immune cell infiltration in breast cancer has led her on the path to really treating a number of patients with immunotherapy in this country. In fact, Shireen Loy was the clinical lead for the first patients who were treating with atezolizumab here in this country. Now FDA and TGA approved many HER2 and triple-negative breast cancers. And of course, now she's the lead for a number of different clinical trials, not only for advanced breast cancer, but also for example, triple-negative breast cancer. If you can give patients immunotherapy, reduce the tumor prior to surgery, you could achieve cure. Really revolutionary stuff. And with this funding and her discovery, she's now been named as the 2021 Frank Fenner Prime Minister in the Frank Fenner Prime Minister's Prize for the Best Life Scientist of the Year. So we support winners. And I thought I'd leave to this one because we've got a very special researcher that we support as well. In the 2021 round of the IIRS, Associate Professor David Gallego Ortega has also been successful in attracting funding through this scheme. And of course, David will tell you all about his research, but basically what David is doing is he's using an external ex vivo model using this Alton technology to really simulate what the patient situation is ex vivo. So he can test a range of different drugs to personalize the treatment for that patient. And I think that's a revolution. We can't wait to see the results of your study, David. So overall, in the last 10 years alone, National Breast Cancer Foundation has invested $120.1 million into 270 world-class breast cancer research projects. What have we achieved? New knowledge. I don't have to tell you all that publications are impact. Over 2,320 publications have been published in that time. 46% of all the National Breast Cancer Foundation grants have attracted further funding. I just gave you an example of Brendan Kennedy, who attracted Medical Research Future funding. We've attracted an additional $211 million of funding for our funded researchers. And it really does have true economic and health impact. 18 intellectual property licensing and incidences. You all know it's not enough to do work in the lab. You've got to get it into the clinic. You have to license that product and change the way we treat patients. So 80 incidences of informing policy and practice, and 395 new national and international collaborations have been made in that time. So true impact. We are absolutely proud to continue to invest in the very best breast cancer research in this country. And I implore you to apply to the scheme, because the more research that we do, the closer we're going to get to our vision of zero deaths. Thank you.
Thank you very much, Sam. That was very impressive to look at the numbers. I mean, obviously, we've known about the NBCF, you know, from a long time, and the work that the NBCF does, not only for funding research, but also to raise awareness about in the community about, you know, how important is research to get to the cure for breast cancer. So it's very impressive to find those numbers and see how much impact really NBCF is doing from, you know, from the Australia side. So it's very proud to be part of it, actually. Thank you very much, Sam. So we're going to move along in the interest of time. And now we're going to move into the two presentations from scientists. We have the overview of the associations that are involved in this symposium from the ASEICA and the NBCF. And now we're going to move again back to Spain to host Dr. Maria Muñoz Cafarel. She is a principal investigator of in the breast cancer group in Biodonostia Health Research Institute in San Sebastián in Spain. And she's going to tell us about new avenues for breast cancer immune therapy. Over to you in Spain, Maria, and welcome. Thank you very much.
Hi, hello. Hi, Can you hear me? Can you see me? Yeah. I'm very happy to be here today. Thanks, David. Thanks, Coral, for the invitation. And I will now share my screen and tell you a bit of what is cancer immunotherapy. Sam has already mentioned it. And a bit as well of the work we do in the lab here in a corner of Spain, in a beautiful city, David can say, which is called San Sebastian. So I'll share my screen and you let me know if it works. Great. Can you see my screen now? Yes? Perfect. So David was saying, I wanted to give you today an overview of what is immunotherapy and how can it help to breast cancer. Okay, let's see if I can move the presentation. Perfect. So as you may all know, breast cancer is caused by transformation of the mammary epithelium, by transformation in this ductal alveolar structure, and it progresses through different stages. Currently, our research, focuses on the breast cancer that has already disseminated to other organs in the body, because as Samantha mentioned, the rates of survival for breast cancer are luckily very good, and thanks for research, but still we do not have effective therapies for the breast cancer that has disseminated to other organs. We offer now mainly palliative care, and this is where we want to focus our research and where funding bodies are mainly focusing their money. And I will give you some numbers that will add on to what Samantha has just mentioned. Around 30% of the breast cancer that are diagnosed every year are already diagnosed as locally advanced. What does it mean? It means that it has already progressed or invaded the associated lymph nodes. 5 to 8% of the breast cancer that are diagnosed every year are already diagnosed as metastatic. And as I mentioned, metastatic breast cancer is the one that has already invaded some distant organs, as can be the bones, which is the organ where most of the breast cancer cells will migrate or will go, but also the brain, liver, lungs, or many other different places. And this is where we want to focus our research. And actually, there will be another 20 or 30% of cancers that will be diagnosed as early, but with time can progress to metastatic or can reappear again in form of metastasis. And this is, as I was telling you, where we're going to focus our efforts. And I will now introduce to you a slide that you may have seen in the past, which is called, sorry, give me a second, which is called hallmarks of cancer. What are the hallmarks of cancer? Hallmarks of cancer are characteristics that every cancer has to progress. There are many different ones, and I won't talk about them today. Some of them are obvious, as sustained proliferation or resisting cell death, which make cancer cells to proliferate forever and to keep growing forever. But I will focus today on these two that are the ones related to immunotherapy, which is avoiding immune destruction. Cancers learn how to protect themselves from the immune system. The immune system, as you know, is there in our bodies to protect us from infections, diseases, and also for cancers. Actually, if it would not be for the immune system, we would have many more cancers than the one we have, because the immune system usually is very effective on destroying every little cancer cell which is around our body. But when cancer progresses and when cancer establishes, it learns how to hide from this immune system. And also another hallmark of cancer that is going to be important for immunotherapy is tumor-promoting inflammation. And I will come back to this hallmark later. But in summary, inflammation is the infiltration or the presence of inflammatory immune cells in the body, in the cancer that can end up being pro-tumor. And what is cancer immunotherapy? So cancer immunotherapy is the name that we give to the therapies that are aimed to help the immune system to fight against cancer. And it's not a novel idea at all. Actually, the concept appeared in the late 19th century when Virchow was the first one detecting that the tumors are infiltrated by immune cells, that he can observe immune cells inside those tumors. And actually, it was in the 19th century when accidentally they made the first cancer vaccine work. Then it's true that there have been like several highlights or several key events in the development of immunotherapy, but it has not been until 20 or even 10 years ago when cancer immunotherapy has really exploded. And actually, it was around 2010, 2014, 2015, when the first immunotherapy immune checkpoint inhibitors that I will explain later actually were discovered. And in 2018, that is not here in the timeline, the Nobel Prize was awarded to the discovery of this immune checkpoint inhibitors. And to understand what is cancer immunotherapy and how they work, we have to understand that tumor progression is a multi-step process orchestrated by many different cell types. And we call these many different cell types the tumor microenvironment. And what is the tumor microenvironment? The tumor microenvironment is composed of every cell type that is present in the tumor apart from the tumor cells. Okay, you can see here a tumor. The tumor cells will be in green, but you will have in the tumor many other cells, immune cells, cancer-associated fibroblasts, cells that form the blood vessels that will play an active role actually in the development of the disease. And they can help the cancer to progress in the primary site. They can also help the tumor to intravasate to the blood vessel and actually to develop or to develop metastasis in distant sites. And in the tumor microenvironment, we found the immune system. And this immune system can have a yin-yang effect, would we say, okay? Because the immune system in principle, as I was telling you, is there to protect us from cancer. And actually there are some immune cells that are made for to directly kill the tumor cells. There are others that what they will do is to present cancer antigens to the T cells. They will act as presenters. They will tell to the killer cells, look, there is a cancer cell here that you have to kill. And this is what the immune system will make in principle, like to protect us from the cancer. But as I was telling you, it can have a yang side because tumors can educate this immune system actually to help the tumor progress. And we will have elevation of pro-tumorigenic and pro-metastatic cytokines. We will have, for example, presence of tumor-promoting macrophages, and then the immune system will help the tumor to develop. So as I told you, immunotherapies are every therapy that will target the immune system in order to fight cancer or to make immune system to work better to destroy cancer. So there are many different types of immunotherapies. we have, and some names may be familiar to use, we have this anti-PD-L1, PD-L1, CTLA4, which is what we call immune checkpoint inhibitors. We have as well the CAR T cell therapy that has been very, very effective in hematological tumors. We still need to work a lot and to do lots of research to make those CAR T cells work for solid tumors such as breast cancer. We have as well vaccines. We have release of cytokines, and all these therapies will work or will target different points of the immune system activation cascade. We can help the cancer cells to be more prominent or to the immune system by showing more what we call antigen. In this way, they will not hide anymore from the immune system. We can as well help the presentation of these cancer cells to the killing cells. We can as well help the trafficking of T cells, which are the killer cells, to the tumor. Or what we can do is to help those T cells effectively kill the tumor cells. And I will now to explain a bit about the immune checkpoint inhibitors, because they are the only therapy which is approved now for breast cancer. Samantha already introduced to you that breast cancer is very heterogeneous. We have very different subtypes, and actually immunotherapy is only approved currently, nowadays, for triple negative breast cancer. It was first accepted to treat metastatic triple negative breast cancer. And I'm showing you here, I don't want you to go through details at all, but those three, in patient 131, in patient 130, and this keynote 355, were the main or the key clinical trials that led to the approval of atezolizumab and pembrolizumab, which are antibodies, which are immune checkpoint inhibitors that are accepted now for breast cancer. And recently, they have been accepted as well for early triple negative breast cancer. And how do they work? Samantha mentioned this concept before. As I told you, tumor cells can educate the tumor cells the immune system to hide for it, to hide from it. And actually they can show these spikes, which are receptors with different names, that bind to some other spikes or receptors in the T cell and put them off, put them to sleep. Like this way, the T cells cannot really kill the tumor cells. Those immune checkpoint inhibitors, what do they are? They are drugs that will bind to these spikes. They can bind to the spikes or receptor in tumor cells or to the spikes or receptors in the T cells. And they will then break this inhibitory signal, and they will make the T cell active, and this T cell will be able to kill the tumors. And in the last 5 minutes of my talk, I will give you a very short overview of what we do in the lab regarding immunotherapy. And in the lab, we have decided to focus on this hallmark of cancer that is called tumor-promoting inflammation, that I already very briefly introduced to you. What is inflammation? Inflammation, as I told you, is the infiltration of the presence of immune cells in the tumors. This is a tumor full of cancer cells in green. And as you can see here, we have also lymphocytes, we have neutrophils, we have macrophages that are present in the tumor. And with this inflammation, where this presence of immune cells is chronic and it's in very high levels, it can be pro-tumorigenic. Okay, and it can be this cancer-promoting inflammation. And as I told you, Virchow was the first one to see those immune cells in the tumor, and the concept is not new at all. Actually, Dvorak, I don't know how many, 40 years ago, he already said that tumors, infiltration in tumors is like wounds that do not heal. when we have a wound, immune system is going to be recruited there to actually close the wound. But if this doesn't happen effectively, and we have a chronic and persistent inflammation there, we can have a problem, and we can have wounds that do not heal, and we will have the opposite effect of what we want from the immune system, which will be here a pro-tumoral effect of the immune system. Another analogy or another way to show you how infiltration, how immune infiltration works, is this analogy of the tree, the rain, and the soil. And in this picture, we will have the tumor as a tree that needs the tumor microenvironment to grow as a fertile soil, okay? The tumor really needs the presence of other cell types to actually grow and progress. And inflammation in this picture will be the rain that helps the tumor to grow. And how is inflammation controlled? Here I will introduce you the cytokines. Cytokines are small proteins or small molecules that act as signal molecules or signal messages. They are the key proteins or the key messages liberated or secreted by immune cells, but also by epithelial cells and cancer cells that will be secreted by those cells and will act on other immune cells or other epithelial cells. Okay, they are the language that immune cells use to communicate with each other, and they are needed in normal physiology. But if they are overactivated, they can create problems that you will all know, such as allergy, chronic inflammation, and autoimmune diseases. And they are very important in cancer as well. While I was in Cambridge doing my postdoc in Nick Holman's lab, my project was aimed at focusing on what is one of these cytokines, that is called oncostatin M, OSM, doing in cancer cells, okay? And by doing many experiments, and after many years of research, we found out that this cytokine in cancer cells activated key processes for the cancer, such as angiogenesis, which is the formation of new blood vessels, and migration and invasion. And when I started my work in Biodonostia as a principal investigator, I decided to study, because I thought we were missing the whole picture actually by focusing only in cancer cells. And I wanted to study what is the role of this cytokine, and we study now other cytokines in the tumor microenvironment. Because maybe our hypothesis was that apart of inducing new blood vessels and metastases, it may be promoting as well tumor promoting inflammation. And this is work that was started by Angela and Andrea, two exceptional, brilliant PhD students when I started my lab. And I will not go into technical details. I will just show you 2 or 3 slides of experiments to show you how we work in the lab. And we use lots of mouse models. In particular, for this project, we have used this mouse model, which is MMTV polyoma T. That is a genetically engineered mouse that will develop spontaneously breast cancer during its life, because it expresses the oncogene polyoma T in mammary cells. And this model is very helpful because it goes from benign lesions called hyperplasia to late carcinomas, and it eventually metastasizes to the lung. And this recapitulates very well what happens in human cancer. So what we did was to cross this mouse model that expresses breast cancer spontaneously with mice that lack oncostatin M signaling, okay? They do not have the receptor, oncostatin M receptor, and therefore they will not have the cytokine. We cross these mice, we follow the progeny, and we follow tumor progression in these mice, in this progeny. And what we observe is that actually OSM signaling promoted cancer progression. These are a representative picture of a mammary gland, of a murine mammary gland, of a mouse mammary gland. You can see here, this is the fat that we found in the mammary gland. This is the lymph node, this dark point. And all these dark nodules are is the tumor that is starting to grow, okay? And this is the nomenclature, the name we use in science. The knockout animals are the ones that do not have oncostatin signaling. And as you can see here, I hope you can see, these mammary glands are less invaded by tumors than the control mammary glands. I will skip this because of lack of time, but I will show you a very interesting experiment we did in collaboration actually with David in Sydney. And it's like science has really progressed, and we have now technical advances that allowed us to really look into the tumors at a very, very small level, no, with lots of detail. And one of these techniques is single cell RNA-seq that maybe Laura will explain later. And it allows us to separate the tumors in maps, which have funny shapes. I actually see a little fish in this map. And each population here, each like group of dots will represent one population one cell population in the tumor microenvironment, okay? We will have the cancer cells, fibroblasts, immune cells here, T cells, B, lymphocytes, myeloids. And what can we do with those maps? We can ask those maps where our protein of interest or where our gene of interest is actually produced. So we found out that the cytokine OSM, and I don't want you to pay attention to the data, but you see here the red points that coincides with this myeloid population. We found out this way that the cytokine OSM was produced by myeloid cells, mainly macrophages in the tumors, and that the receptor OSMR was mainly expressed in cancer cells and fibroblasts, that were the cells able to respond to OSM. I will skip the next slides for lack of time, and I will just end up with the model after many years of experiments we end up with, okay? This is a picture summarizing 5 years of research, and our model, final model to explain how OSM promotes breast cancer is what we call this ménage à trois, that is activated by OSM, but the cytokine OSM, and it's a ménage à trois between macrophages, which are the immune cells that secrete OSM in breast cancer, cancer cells that will be here depicted in blue, and cancer-associated fibroblasts that will also respond to this cytokine. And all of these 3 cell types will activate a pro-tumor OSM loop in breast cancer. And I will end up my presentation here just by showing you the people who actually did the work. I am very grateful to work with these extremely intelligent people and very nice. And I will be very happy to take questions at the end. Many, many thanks. Bye.
Thank you very much, Maria. That was great. It was a very interesting overview of cancer immune therapy and how your research is adding a lot of value into that, kind of finding new cytokines or new proteins that can be used in the future for designing new therapies in that space. As we've seen, checkpoint inhibitors, for example, are actually quite a promise for the treatment of cancer. Moving along in the interest of time, we are now have our last presentation from a researcher. So we have here with us, back to Sydney, we have Dr. Laura Rangel Sanchez. She is a research associate at the School of Biomedical Engineering at the University of Technology, Sydney. And she's going to be talking about the funded project from the National Breast Cancer Foundation that Sam was alluding before, which is called Personalized Medicine Using High-Fidelity Patient-Derived Tumoroids. Over to you, Laura.
Thank you so much.
Thank you.
Well, thanks for all. I'd like to thanks all the organizers for giving me the opportunity to talk in here about the project that we are currently working on, which is using high-fidelity patient-derived tumoroids for personalized medicine. And I guess some of the things that I'm going to talk about have been already discussed, but I think it's important, to stress the importance of breast cancer research and what we know and what we could do. So, as Sam has mentioned before, breast cancer is the second most commonly diagnosed cancer in Australia. This means that over 20,000 Australians will be diagnosed of breast cancer each year, and in Spain will be over 34,000 women will be diagnosed this year of this disease. This means that between 1 in 7 and 1 in 8 women will develop this breast cancer during their lifetime. And although not all news are bad, and early detection programs have helped a lot in improving the patient survival, breast cancer still remains the second most cause of death from cancer in females. And it accounts with 608,000 deaths per year globally, which is a lot. But I'd like to talk from the very basics. What is cancer? And cancer was defined by the Greeks as carcinos, then was translated to the Latin, that was cancer. That's why it's called cancer, which means in Latin crab. This is because of the shape of this disease, that was this finger-like spreading projections that look like a crab. Breast cancer all cancers start when there is a genetic alteration in a healthy cell that promotes the rapid division and proliferation of the cell. And in later stages, these cells will recruit the blood vessels in order to get more oxygen and nutrients that will support their proliferation. At the very late stages, these cancer cells might migrate and they might invade another sites of the body, promoting secondary tumors, which is called metastasis. But as Maya already has shown, tumors are not just made up of cancer cells, but also by other a myriad of cells that form the tumor microenvironment, which are the immune cells, the fibroblasts, and there's also some supporting protein architecture that supports all the cancer growth and metastasis. But this is also important for a response to therapy, as Maria has mentioned before. And this is kind of like an overview of what is breast cancer. Breast cancer is just not one single disease, but it's a compendium of different subtypes. These subtypes have been classified in 4 main breast cancer subtypes based on the expression of 3 different genes, which are estrogen receptor, progesterone receptor, and HER2. This define these 4 luminal A, luminal B, HER2 positive and triple negative breast cancer subtypes. And just to get an idea of the complexity of this disease, as an example, triple negative breast cancer is further subclassified in 6 more molecular subtypes. So you may imagine that this breast cancer is a very wide term. And it's important to stratify the patients in order to deliver the best, because of the microphone, to deliver the best therapeutic approach and achieve better clinical outcomes. But this is not as simple. And in addition to this, this interpatient or intertumor heterogeneity, it has been now shown that within the same tumor, there is what is called intratumor heterogeneity. This means that the tumor is very heterogeneous, and if you zoom in, you can find different clones of cells, of cancer cells, that are very different between each other. And they might respond differently to therapy. And actually, this is a key for therapy failure. Why is this? Well, I'm going to show you an experiment that we recreated in the lab. This is done in mice. So if a population of cancer is very heterogeneous and you treat it, you might see, as you can see in the purple line, firstly, you might see a regression of the tumor. But this doesn't mean that all the cells are dead. So this, if the tumor is very heterogeneous, some cells can survive, which we call the resistant cells. And these cells will regrow. And even though we treat them again with a chemotherapy, these cells might resist. And this is really bad because these cells are now very aggressive, resistant to therapy, and they might metastasize. So traditional standard of care therapy usually tests the overall characteristics of the tumor, and then based on that, they deliver a chemotherapy. And this might be okay for some of the patients, but if the patients are not properly stratified, for some other patients, this therapy, it might not only work, but it might also cause the metastatic spread of the cancer and making these tumors even worse. Thankfully, now personalized, the medicine is trying to shift into a more personalized approach, in which we are trying to stratify better every single patient, and then based on the intrinsic molecular characteristics of each tumor, deliver the best therapy possible. This, of course, not only enhances the treatment efficacy, but it also lowers the side effects, and of course it reduces the cost in the health system. But to deliver a proper personalized medicine, we need to clearly understand the pathology and the molecular underpinnings of each tumor. It is also important to develop new targeted therapies and novel drugs that can target these cancer therapy-resistant cells. And of course, we need drug screening systems that can predict how the patient will respond to that therapy. Currently, only 1 of every 10,000 compounds that are developed in the industry will reach the commercial stage. And this is due in part because of there are not reliable drug screening systems in the preclinical stages. So we have been traditionally using these 2D cell culture models for drug screenings, where we plate the cells in a flask. And this is amazing because it's very cheap. You can try test a large number of drugs. But as you might know now, tumors are not just made up of cancer cells, but also other non-cancer cells that help in this tumor progression. And these systems, they are very homogeneous, so they don't recapitulate the patient's tumor complexity. Therefore, they are not clinically predictive. There is another alternative, which is the use of animal models, that while they do recapitulate a patient's complexity, they are slow, they are very expensive, and of course, they present ethical concerns. So in order to address this problem, David Gallego's lab invented this ALTEN, or alginate-based tissue engineering, which is basically, he gets small pieces of tumors, and as you can see here, and he can embed them in a medical grade hydrogel that you can see here in this image, and maintain not only the structure but also the cells alive for a long time. This has the advantages of the 2D models because they are high throughput, so we can test a lot of drugs, but also has the advantage of the animals because they maintain the complexity of the original patient's tumor. So in addition, we are able to automate this process using microfluidics. So we can generate hundreds of these tumoroids, I'm going to call it for now, very quickly. And and we can control also the size. So it's very reproducible, and we can really fine tune as we want. So now, in a project that is funded by National Breast Cancer Foundation, as Sam has already mentioned, what we want to do is taking breast cancer tumors, embed them using our automated medical device, and culture them in plates in absence or presence of different therapies, mostly chemotherapies. Then we will, based on the sensitivities that we get, so which drugs are more effective on these tumors, we will be able to predict for which drug this patient will be more sensitive, the tumor. But we are also going to do single-cell RNA-seq, and Maria already has talked a little bit about that. And we will do this in order to characterize the molecular pathways that are inducing these cancer cells to resist to therapy. So we then will define the molecular targets of these, what makes it special to these cancer cells. And in the future, this could lead to the development of new anti-cancer drugs. So now let me explain you how we are going to define these molecular mechanisms of chemoresistance. And so I'm going to explain you a little bit, as David has suggested, the smoothie effects, in which the single-cell RNA-seq is based. So I want you to see these fruits as a tumor, but as you might imagine from my previous images, tumors are not like this. They are more like this fruit salad. They are complex and they have very intricate structures and interactions between different cells. So it's kind of difficult to get to know what is going on in there. So traditionally, all genomics, transcriptomics, when we want to know what are the genes that are being expressed in this fruit salad, we will smash it, and then we will get average signal, which is the smoothie, that, as you can see, is pink because of the strawberries, and it might taste like bananas, but maybe you don't know that there is a kiwi over there. So if you only look at it, you might only think that it's the strawberries. So the signal is really masked, and it's difficult to know what is actually going on in these tumors. What single-cell technologies does is separating by features each of these cells. And now it's much easier to go to what are the molecular characteristics of this each of these cells. So if now we translate that into a tumor setting, we have here the tumor with the tumor microenvironment. And what a single-cell resolution does is separating the different cells. So you can see the fibroblasts, the cancer cells, the blood vessels, the immune cells. And now we can test what are the specific characteristics of each of these cells. So this doesn't look like this in a lab. It looks more like this. So here you can see the fibroblasts, the blood vessels, the cancer cells, and immune cells, where each dot is a single cell. And you already can see that cancer cells are quite heterogeneous. So in order to test whether we could combine ALTEN with single cell technologies to unravel mechanisms of chemotherapy resistance, we took mammary tumors, embed them in the ALTEN medical device, and culture them in presence or absence of a common chemotherapy used in breast cancer. Then we performed single cell RNA-seq and got this beautiful map of the cancer cells. And you can definitely see at least 3 different populations that if you go to the trajectory map, you can see that in the non-treated cells condition, we have like 2 states of the cells, and one of them, when after treatment, it disappears. The yellow population disappears, while it appears this green new population. However, if you look to this red population, it seems that it doesn't change in the presence of chemotherapy. We call these cells the resistant, chemotherapy resistant cells. So now we can go to each of these dots and check what are the molecular characteristics of each of these cells. And I'm not going to go through it in much detail, but I can tell you that we validated a few of those highly expressed genes of these therapy-resistant cancer cells. And we could validate this in a probe that is usually done in hospitals when they check for a patient's tumor pathology. And as you can see, for one of the markers that was highly expressed in this therapy-resistant population, that you will see in green, after chemotherapy treatment, the live cells are actually the ones that are expressing this marker. Why is this important? Well, this marker can be used now as a marker to predict a therapy response. When we get the biopsy from the patient, we know that he or she has cells that are expressing this marker, then we know that we cannot use this chemotherapy anymore. And additionally, we can now develop new anti-cancer drugs that will target these cells and probably will kill these cancer-resistant cells. And just my last slide, how we want to implement our ALTEN in the clinical pipeline. Usually physicians will take the tumor biopsy, and up to now they will send it to pathology, and they will have an overall checking to know what are the main characteristics or the subtype of the cancer. What we would like in the future is to get part of that biopsy and culture them in the presence of different drugs, different combinations of chemotherapy, and with this data, inform the physician which will be the best therapeutic approach for this particular patient. But in addition, we want to perform single-cell RNA-seq, and this is really great because we can now define novel mechanisms of chemotherapy resistance that could lead to the development of novel anti-cancer drugs that, of course, can be tested in our ALTEN device and eventually go to the clinic. That's all from me. I'd like to thank to everyone in the lab, the cancer fighters, you can see, David Gallegos lab and Fatima Valdez lab, and of course the funding members, National Breast Cancer Foundation, NHMRC, and Fundación Ramón Areces. Thank you for listening.
Thank you very much, Laura. That was great. Thanks for the update on what is going on in that preclinical testing for using the high fidelity tumoroids. So I guess this concludes our talks and presentations. So now I think that we can have a few minutes. We are a little bit late on time, but we can have a few minutes of questions if there is any question in the audience, or in the audience, or maybe online. Please go ahead and ask questions. So there's Coral. Yeah.
Online, I have some questions from our guests online. The first one is for Samantha. Thank you so much. What are the key factors to finally choose allocation of funds, effectiveness, time frame to be used in the general public, or?
Yeah, so we have a fairly strict range of category descriptors. So when we, at the moment, our investigator-initiated research scheme, we ask applicants to apply for funding for up to $1.5 million in funding. We have a funding cap of 1.5 per principal investigator. We then allocate those applications among panels based on the total level of funding. So the idea is that if it's a small grant, we try to compare small grants with other small grants. A larger grant will be compared against larger grants. We then ask our reviewers to review, and they review based on very strict category descriptors for the grants. So our 4 main categories are obviously scientific quality, team capacity. So scientific quality being the quality of the application, and that must show feasibility. You've got substantial preliminary data that will give you the feasibility to conceive the approach. It has to be a very good idea, obviously, that's going to make a substantial impact on breast cancer. Team capacity. So we absolutely strongly recommend that all researchers at all career categories apply, because we don't want just senior investigators to get their grants. Of course, it's important for senior investigators to apply, but we really encourage the mentorship of early and mid-career researchers. So, for example, you can have a very junior researcher that can lead an application and be supported by a very strong team, and that will still give you a strong team. The third category descriptor is significance and alignment with NBCF goals, and that is ultimately our vision of 0 deaths. And finally, we have a category of innovation. So that the grant must be truly innovative, but we're not restrictive. It doesn't have to be innovative across all areas. It doesn't have to be necessarily innovative in idea, but it can use an innovative technique to address a really obvious question, or it can be innovative in idea and use some very stock standard gold standard techniques that have been around for 50 years. So we're fairly liberal with that, and we asked our reviewers to score on that. In addition, our research is absolutely applicable to the end user, obviously. So we ask our applicants to apply and have consumers involved in the research from all the way from conception of the idea to all the way of dissemination of outcomes. And so we also ask our researchers, and we score the application on the level of consumer involvement that's involved in that application as well.
Thank you. Thank you so much. A very complete and comprehensive answer. I must add that this conference is being followed by fellow researchers, but also non-researchers, as myself. And this is one of the key issues to promote in the general public, as we can say, this field of research, and to do everything we can to try to help. So there goes my second question. This is from Instituto Cervantes. What can we do? What can, on the one hand, general public, and on the second hand, public institutions, what can we do to help in this area, to help in the research? So this is a general question for you three and for also our colleague in Zoom. Here, I can give you mine.
So maybe I can start with that question. I think, I mean, obviously, we are all here today. I think it's because this is important. So, of course, we are doing. I think that every player needs to play its part. Of course, we have the researchers working in the lab, long hours and trying to, answer very, very difficult questions. We also have as Samantha just said, the end users, that they have an input because they're obviously aware of what is the demand, what is important for the real world, on where do we need to, I mean, everything is important, but where do we need to really focus our studies? But also I think the community, you know, like we have, for example, from the point of the donors from supporting the National Breast Cancer Foundation, this is actually critical to for people to understand what foundations or associations, non-for-profit organizations such as NBCF do, and we have an excellent group of people trying to recruit all those fundings, working really hard to get the money that the researchers need to, I guess. And Sam was offering a few interesting numbers in her presentation, top up all those government fundings that actually not enough for the work that needs to be done. But also I think that we hear today about ASEICA, which is also another association. It's an initiative and it's being led by researchers. They have now ASEICA Joven, ASEICA Med, and a number of those ones where Tony probably can tell you a lot more, but really bring together the collaboration and the kind of interaction between researchers. And lastly, I guess, I think events like this one. So events like this one are important. So we are not working in isolation. So we're actually working together and then, promoting, and so people is aware of what we do and, what is the role that everybody plays in this kind of situation. So we're all in the same boat, trying to get the same aim. I think it's a very interesting and inspirational aim for the National Breast Cancer Foundation, the 0 deaths. I think that's towards we are all working. We all have, unfortunately, in our family, friends, and close people that is being affected by breast cancer or all the type of cancer. So I think the research is actually quite important. So I think it's just channeling that energy towards, like putting the energy into finding those cures. I don't know if you want to add anything. You have a microphone.
Doesn't matter. So yeah, I think adding to that, I think we as researchers, we have also the responsibility to reach the lay people and make them understand that what we do is important, that it's going to affect them directly, and just convince them that what we do is important, and probably like bringing more education about it. But we as researchers, I feel like we have this responsibility to make it in a way that everyone can understand it, because many times we just talk about things that is like sales. I don't know, where are the sales? What are you talking about? So I think we also have this responsibility to be able to reach the lay people so they can understand and help us to help them.
And just from a National Breast Cancer Foundation perspective, obviously our researchers are key to getting those messages out. And as I said to you before, we're a 100% community funded organisation. We do not receive any money from the government. So we just want you to keep donating and get the message out there that dollars means research, and research saves lives.
Sorry.
If the people from Spain.
Oh yes, sorry. Of course. I'm not sure if Tony or Maria want to add anything from Spain to that question.
Hi, hello.
Yeah,
Tony,
we can hear you.
Yeah, Tony had to leave, but I'm here. I mean, as Tony said, I work in ASEICA as well. I don't know. I agree with the three of you. I think it's very important that we researchers made us easy to understand to lay people, and that we work together. For example, now I'm very happy that I work in a hospital and we now have a patients committee, and they actually participate in the decisions that we take, in the clinical trials that we participate. They ask us questions, they ask us training. And I think that only in a close interaction between science and society is how we are going to make this to work. I mean, if we have to be thankful to the pandemic in some way, it's because now people know what is a PCR. And yeah, so somehow like science has become more popular. So I would say that please keep this positive attitude to science. Science, as Samantha was saying, science cures. So I think this is something that we have to do all together. Thank you.
Thanks, Maria. That's great. So I'm not sure if there is any other question. I mean, we are also running out of time. We're a bit late already. But look, I would like just to close the session and really thank Coral and the team for hosting us here in the Instituto Cervantes in Sydney. It's great to be here, raising awareness for breast cancer and then trying to communicate and help a little bit to that kind of bridging that gap into the communication from the researchers and the actual public. And also like to really thank the four speakers, the time from the ASEICA, Tony, for giving us the overview of what is happening at that side of the world, for Samantha Oakes as well from the National Breast Cancer Foundation for her time and, kindness to be here today and telling us about, the wonderful work that NBCF is doing. And also Maria there in Spain, very great to see you and to see, how your work is developing. Really looking forward to see what are you coming up in the next few years and what are your discoveries. And also Laura for telling us all about the new research and the new projects that are happening in the personalized medicine space. Thank you very much, everybody. And thank you for everybody that has been here today and online in Spain, in Australia. It's been a pleasure. And have a good day and good night in Australia. Thank you.